Today we will be presenting our CA9 protein project by Charmaine Vergara, Nayeli Vega, and Vivian Din.
Hi, my name is Nayeli Vega and I'll be presenting the abstract and the background. Our enzyme is carbonic anhydrase 9, which is an enzyme in charge of regulating the pH in cells, but when there is no regulation, it will favor the conditions for cancer cells to survive, grow, and spread in low oxygen conditions. Based on this, a Western can be run to prove the expression levels of the enzyme in different conditions to verify its presence and whether its expression correlates with pH regulated, a normal cell, or cancer cell behavior. Finally, we will compare our enzyme to trypsin. Our enzyme does not directly make cells detached or round like trypsin would, but it does affect how cancer cells move and spread by creating the conditions for both angiogenesis and metastasis to happen. So a little bit about our enzyme, its upregulation in hypoxic tumors is involved with their survival, growth, and metastasis by helping tumor cells adapt to acidic microenvironments. It also promotes cancer by supporting cancer cell survival, maintaining intracellular pH, and facilitating acidic waste, allowing them to thrive, evade apoptosis, and resist treatment. Our enzyme also teams up with other proteins from hypoxia inducible factors, mainly alpha-1, to help survive and grow cancer cells in low oxygen conditions. So our enzyme works at a gene level, influencing the expression of other genes indirectly by altering the pH of the tumor microenvironment, interfering with pH enzymes and signaling pathways involved in cancer progression. Finally, our enzyme does not directly degrade proteins, but it does indirectly contribute to the activation of acid-dependent proteins, which can facilitate metastasis by breaking the intracellular matrix.
My name is Vivian Din and I will be presenting the experiments and figures. So for the technique, we chose to do a Western blotting because our protein is involved in cancer by regulating gene expression. And we can use a Western blotting to identify and quantify CA9. We will be testing for CA9 expression in normal cells and cancer cell pH conditions. In normal cells, we would expect to see low CA9 expression, and in cancer cells, we will expect to see high CA9 expression. So, like I said in the last slide, we're going to have a positive and negative control to compare our cells of interest to in our Western blotting. The positive control are HeLa cells treated with hypoxia, and our negative control is HeLa cells treated with normoxia, which is around 21% oxygen, and hypoxia being around like 1 to 2% oxygen. So moving on to the Western blotting procedures, since CA9 is a membrane-bound protein, we need to lyse the cells with a RIPA buffer containing protease inhibitors in order to release the proteins from the membrane. Next, we're able to separate the proteins on an SES page gel. And then in step 3, we're going to transfer them to a nitrocellulose membrane to make the cells more accessible for antibody binding in step 4. So now we're going to incubate the cells with a specific CA9 primary antibody, and we're going to be using the mouse anti-CA9. And then in step 5, we're gonna apply a secondary antibody conjugated to horseradish peroxidase in order to visualize the protein bands using chemoluminescence detection in step 6.
Hi, my name is Charmaine, and I will be presenting the relationship to trypsin-treated cells in angiogenesis. So carbonic anhydrase 9 does not cause cells to look like cells treated with trypsin. CA9 promotes epithelial to mesenchymal transition, or EMT, which results in a spindle-like morphology, unlike a round and detached state seen in trypsin-treated cells. EMT is facilitated by a loss of cell-to-cell adhesion when tumor cells become invasive. As for angiogenesis and metastasis, this invasive morphology allows cancer cells to invade nearby tissues by facilitating interactions with endothelial cells and blood vessels. Hypoxia-induced expression stimulates vascular endothelial growth factor, or VEGF, which forms new blood vessels. The spindle-shaped morphology also allows cancer cells to migrate around the extracellular matrix and invade surrounding tissues. The acidic microenvironment and mesenchymal morphology promotes survival at the metastatic site. As seen in Figure 2, the protein links overexpression with migration and invasion in a molecular process that occurs within the cell. This can be seen when CA9 induces oral cancer cell motility. And here are the references.